And we have an awesome lineup on chromosome dynamics, but first we're going to start with our very first award, and Phil Heater is going to introduce this. It's the Winge-Lindegren Award.
Can you change the slide to my presentation, please? Nope, there's one from mine that I had loaded. Or should we just forget it? I don't need the slide, it's fine. So, anyway. So this year's Winge-Lindegren address is being given by Rodney Rothstein. And as many of you may know, the Winge-Lindegren Award is meant to honor a leader in the yeast genetics field and gives them the opportunity to give a thought-provoking perspective on pretty much anything they'd like to talk about. So I'm really looking forward to hearing this. And I just would say a couple of things about Rod. Next slide, please. Is that I think there are three things I'd just like to say about Rod. One is he is the quintessential yeast community person. He's been contributing and participating in a very good way over 40 years. So he's a guy who comes to the meetings, participates, exchanges ideas, is a real role model for young investigators, and really has contributed greatly to the community. He's a great yeast person. The next slide shows you this. This is the first meeting that Rod went to in 1972 in Seattle. And if you go one more, we blow up the far end, and there he is at the end, standing right behind Herschel Roman, that the young people will not have heard of, but is an iconic yeast geneticist from the old days. And he's actually been at every North American yeast meeting since 1972. He might be the only person who's done that. So it's pretty impressive. So the next slide. The other really important, again, is that, could you go ahead, please? One more. Is Rod's groundbreaking science, and I think everyone's aware of this. He's been studying basic mechanisms of genome stability, DNA repair, recombination over his career. And of course, he pioneered the use of recombination to edit genomes. So he was the first to show that double-strand breaks, really enhance the probability that DNA fragments transformed in yeast would enter into the genome at a homologous site. That was the basis for the double-strand break repair model, together with Orweaver and Shostak in a classic paper. And the one-step gene replacement method really has transformed the field in terms of what we can do in gene knockouts. It was required to do, for example, the entire systematic knockout collection of the yeast. So we're really indebted to Rod for his contributions. And then the last thing I'd say, next slide or next thing, is he's a great guy. He has this kind of Johnny Cash look, and in honor of that, I actually have a black polo shirt on. And as predicted, he's here with a black polo shirt, so that's great. So this is my honoring you. He's a great guy. He's just an outstanding mentor, a lot of fun, and he's been just a great person to have in the community. So we have a little gift for Rod. I'm going to call it the Winge-Lindegren ice bucket, which is inscribed the Winge-Lindegren address, etc.
Where's the gin?
Exactly. This is going to have ice cubes, because he's a big fan of gin and tonics, scotch on the rocks. Anyway, there you go. So
thank you very
thanks
much.
A lot.
Well, thank you. I'm sorry we're a little late, and I'm going to go really fast. So now you put the gray one on. Thanks. And let's go to the next slide. I'm not sure. No, mine's not connected that way. All right. So this is who's talking. Next slide. In case you're in the wrong room, this is all the other places. Okay. So you can figure that out. Could I have the next slide? So I'm delighted to give this talk. I mean, first of all, thanks to the community for honoring me with this award and giving me this great ball. And Winge and Lindegren were two really giants in the field of yeast. And over the years, we've heard different things about them. One is, I don't know if you've ever heard how to pronounce Winge's first name. It's Oiving. Oiving Winge. He's from the Lower East Side in New York. and Carl Lindegren. Well, Winge was actually important at the Carlsberg lab in Denmark. And one of the nice things about the Carlsberg lab, Carlsberg, you know, they make beer. They decided that they should study yeast. And that was, in fact, what's been going on in that lab for the last 100 years or more. And it was very foresightful of that company. And Winge ran the physiology department for many, many years, and basic research was what they were all about. And in his lab, they discovered, in fact, that yeast would mate, that there was a homothallism gene called the D gene, and Lindegren at the same time was doing yeast genetics. Lindegren's lab, Lindegren with his wife, Gertrude, actually found the mating types, mating type A and alpha, which were plus and minus first, by the way. But later were just called A and alpha. And then of course, a lot of other geneticists jumped on board, and of course, that's what's gone on. The yeast genetics started from these two fellows, so it's a great honor for me to get this award. Now, you saw a picture, if I could go to the next slide. You saw a picture of this meeting that I was at in 1972, and the next slide just shows a little bit more close-up, and the next slide shows it up even more close. And I'm not going to go through all the people's names. I've shown this at meetings before. But go to the next slide, and Phil took the same one. Go next. There's the picture of me with Herschel. And Herschel was my real hero. He's my intellectual grandfather. Mike and Shelley Esposito, who I trained with, were his graduate students. And so this meeting in Seattle was kind of old home week for a lot of people, and it was really a really great thing for me. And I also took my movie camera. Now, there were movies, not videos in those days. So the next slide actually shows the movie that I took at this meeting in 1972. And I thought you could hear it, but sometimes you can hear. Anyway, I labeled some of the important people. So some of you might know these names, some of you might not, but we're all getting ready to line up for that picture that I just showed you. And Seymour Fogel, a really important person in the recombination field. John Pringle, who I saw walking around, so he might be here. Yeah, there he is. There's Herschel on the right and Robinow on the left. These are, again, really important guys in the early days. And Don Hawthorne, another one. And you'll see as we go through Fred. I worked with Fred, Mike. Ira was at this meeting. That's when I first met him. Lena Hereford, Shelley, my other advisor. Peter Moens, another yeast cytologist. Tom Pettis. That's not Ron Butow. Haber, Ben Hall, Pete McGee. Yeah, you'll see. Mike Smith is in it. John Warner, Bob Mortimer, Jim Hopper. Jim somewhere here? I know Anita's here. We've got her. There she is. Lee Hartwell, Dave Thomas, John Pringle, again, Seymour. Now, what's interesting, this meeting was in 1972. There were two people who won the Nobel Prize from this meeting. One was Lee Hartwell, and the other is Mike Smith, who's walking off on the side there. So that was kind of cool. All right, he wasn't walking off the bridge. All right, that was my first yeast meeting. Now, the next slide, actually, I thought this would be another nice thing. I took videos of my lab. Oh, that's me, by the way, in 1973. I thought you'd get a kick out of that. Anyway, this is Mike and Shelley's lab in 1974. Okay, so there's my bench. There's my desk. That incubator was only for sporulation because we didn't want to inhibit the spores from forming with other cells. Anyway, there's our high-end microscope, very, very high-end. This is our high-end refrigerator. Looks familiar, doesn't it? That's what's so... Okay, now we're going into the cold room. Actually, it was a really small cold room because it was in Mike and Shelley's lab, but there's the cold room filled with plates and centrifuges and everything else. And there's the dissecting scope, okay? Now, this scope was set up by Mike Esposito and me. By the way, notice it's for left-handed people, because Mike and I were both left-handed. And now we switched to a new lab. This is my lab now. You notice there was a computer on the desk. Oh, by the way, the microscopes have gotten a little bit more high-end. Plates are still in refrigerators. Cold rooms still are cold rooms, and they're full of plates. So not that much has changed, all right, in the ensuing years. Here's the dissection microscope in my lab now. By the way, notice it's set up for right-handed people. I don't dissect anymore, I guess. Microwaves didn't exist in 1974. PCR machines did not exist, and neither did Singer robots. So those are kind of the new things that have shown up. Anyway, so that's, I thought you'd get a kick out of that. All right, so the next slide is another thing that I think a lot of people want to know something about. What is this strain, W303? All right, I'm not going to tell you everything about it, but if I could have the next slide. So when I got to Mike and Shelley's lab, they gave me two strains. They gave me this D gene strain. So D gene is the homethalism. That's HO, by the way. And I crossed it to S112. By the way, it shouldn't be called S112. S112 means it was Shelley Esposito's 112th strain. It really had a name from Mortimer, but it's lost. I have no idea what it was, sorry. But that was my first cross, W1. So why do I use W for my crosses? So now remember, in 1970, by the way, I did my first tetrad in October of 1970. When everybody called their crosses either X, Y, or Z. Okay, X for cross, Y because somebody said, all right, somebody took X, Z because somebody took Y, so I said, all right, I'll go back one letter and do W. So that's where W came from. So so I made strains, they could sporulate well because that's what I was studying. And I moved to Fred Sherman's lab where I did my first postdoc. And in Fred's lab, Fred studied a strain called D3113A, maybe some of you know about it. It turns out that strain's disomic for chromosome 12, stable disome. But anyway, I got some good genetic background out of Fred's strain. Then the most important strain that was introduced into the W303 background was LL20. LL20 was from Lester Lau, LL. Lester was a rotating graduate student in Jerry's lab, Jerry Fink's lab. And he made the Lu2 double and the His3 double. And so that cross went into my genetic background with suppressors and good sporulation and Gal plus, by the way. Next slide. So then I eventually got a strain called W301-18A. Okay, and it was a really nice strain and it transformed really well. But I wanted an A and an alpha derivative of it, so I transformed it with the HO plasmid. That was something that Ira's lab, Rob Jensen from Ira's lab, cloned the HO gene on a plasmid so you could introduce it into a strain, change mating types, make it diplodize, dissect a tetrad. In tetrad number 1, which I labeled 1A, B, C, and D. By the way, capital letters. Anybody who publishes W303 1A with a small a doesn't know anything about yeast genetics, okay? It's capital A, all right? Anyway, tetrad 1 had 1A, mating type A. Tetrad, the second spore of tetrad 1 had the alpha mating type, so that's why you get 1A and 1B. Okay, now, Hannah Klein, my friend, discovered that W303 had a RAD5 mutation. So in the next slide, what we did is we converted W303 to RAD5+. And if you want W303 that's RAD5+, you ask for 1588 or any derivative thereafter. Or you ask for W9100. Actually, I wanted to call it W9090 because it was 30 times better than W303. But unfortunately, that number was taken in the lab. Anyway, this strain has a large copper array, and so it can be used in studies for copper sensitivity. And the last cross that was made in the, oops, not next slide yet. Go back one. One more, forward. Yeah. So the last cross that was made in the lab was 11,222 and counting. Okay, so the next slide, just a little bit, you heard from Phil a little bit about double-strand breaks, and I just wanted to, again, tell you a little bit about the kind of the beginnings of this. When Jack and I were postdocs, Jack Shostak and I were postdocs in Ray Wu's lab. So that was another lab that I went to, but Ray Wu's lab was not a yeast genetics lab. Ray Wu's lab was a lab where we learned how to do cloning and sequencing. And the reason I went there is because of that fact. And Jack Shostak was a graduate student with Ray, and Jack was working on trying to clone the cytochrome C gene from yeast. And again, I won't go through all of that history. But I knew about yeast genetics, and Jack knew about plasmidology, so we exchanged values, and I taught him a little bit about yeast, and he taught me a lot about plasmids. And we then, because at the same time, Jerry's lab, we had where Al Hinnon and Jim Hicks were developing yeast transformation. So we had the ability to now introduce DNA molecules into yeast. And so what we found, and you almost all know about this, but double-strand breaks are recombinogenic, and I won't, again, go through all the details. But Jack and I started that work when we were postdocs, and then we went off separately to start our own labs. Jack moved to Harvard. I went to the New Jersey Medical School. Okay. Jack's first graduate students were Terry Orweaver and Andrew Murray. My first graduate student, I can't remember her name. All right, anyway, I'm sorry, Carolyn, I do remember your name. But she never published a paper. Maybe, and it was more my fault than hers, okay. But anyway, luckily, Terry was one of these amazing people, and when she was in Jack's lab, we continued to study this whole process of plasmid integration. And so I had a wonderful collaboration with Jack and Terry Orweaver, and then we actually published a paper with Frank Stahl outlining how we think double-strand breaks are repaired. If I could have the next slide, I think it actually might show how we think this happens, et cetera. And again, I won't go through all the details. So I've been interested in genetic recombination for a long time. I think a lot of you know that. If I could have the next slide, please. So the other thing that we noticed, and Phil made a comment about this, is we noticed that when you do these plasmid experiments, you could have the ends pointing in toward one another, and then you'd get gap repair, et cetera. Or you could have the ends pointing out, and then you'd get gene replacements. And so one-step gene disruption was born, and that was really important. Now, where did this come from? Where did the idea of doing one-step gene disruption come from? It came from bacterial transduction. So as a graduate student, I studied all kinds of genetics and all kinds of recombination genetics, and the lab across the hall from us was Arnold Raven's lab, a bacterial transforming biologist, I guess I'll call him. And in that lab, I learned, of course, that when transposons hop into genes and they disrupt them in one strain, you could take DNA from that strain using a transducing phage. How many of you know what transduction is? Anyway, and you could move it into another strain. And so what you were doing is you were taking the DNA that was disrupted from one strain and just moving it by genetic recombination into the other strain. So when cloning came around, we realized that we could just make transducing fragments, and that's where one-step gene disruption came from. So again, think about it. It's all about basic science. It's all about understanding how basic processes work. And we didn't learn it from yeast first. We learned it from bacteria, where we learn a lot of things. All right, next slide, please. So then we went ahead and we said, let's look at genetic recombination in living cells. That's something that we got very, very interested in. And again, I won't go through all the details, but we developed methods so that we could basically tag genes in situ, so they had their normal promoters and normal terminators, and they would be tagged with fluorescent proteins. And so RAD52, isolated by Mike Resnick when he was a graduate student with Bob Mortimer in the late 60s, was the first gene that we tagged. And Michael Lispy came to my lab and started all this work with us. And the next slide shows what we found. We found, of course, that a recombination protein was in the nucleus. We found that if you irradiate cells, the lower panel shows irradiation. You can see that with 200 gray are 20 double-strand breaks per yeast nucleus. You get foci. the Rad52 protein, which is all over the nucleus now, goes and makes a focus. Now, there are two things to notice here. One is that there's some cells in the bottom that don't have, this tells me I have 9 minutes. Oh, never mind. All right, that's okay. Some of the cells that don't have foci because those are cells that are in G1, and G1 cells won't make recombination centers. G1 cells are not going to undergo recombination well. The other thing that you might notice is that even though the cells have 20 double-strand breaks, there's only 2 or 3 foci at most, usually 1. And that's because multiple double-strand breaks can come to one focus to get repaired, and the cell has to sort out all of this. And that's an interesting process that we don't understand all about yet. And the other thing to notice is even in the absence of any irradiation, 0 gray, no irradiation, you see a cell with a focus. And it's always, if you look over on the side, it's always a cell with a bud, i.e. a cell in S phase, a cell that's replicating. So during normal S phase, chromosomes break, they need to get repaired, and the recombination machinery shows up. All right, so the next slide shows what we did. We started tagging a lot of proteins, pushed the button again, and what we did is we labeled a chromosome so that we could make a site-specific double-strand break. Okay, so we took advantage of the fact that you could put a multiple tandem array. Andy Belmont showed us how to do this, and next to it we put a nuclease cut site. And we showed that, in fact, the first enzymes that recognize double-strand breaks are part of this MRE11 complex. Again, details you don't have to worry about. But they recognize that there's damage. The cell tries to repair it. Rad52 comes in second. That's what this movie shows. Okay. But the main thing is, before I showed you that you irradiate cells, you see a focus. This is an experiment where we made a break, and we saw the focus at the break. That was the first demonstration that, in fact, breaks and foci really were at the same place. So the next slide just shows a little cartoon of what we think is going on, and that is that ends get broken, the cell makes a decision, should I just glue the ends together, what we call non-homologous end joining, or should I use the homolog or the sister homolog to undergo repair? And the answer is, once that decision is made, then there's a whole choreography that takes place, and that is that single-stranded DNA gets formed, and that triggers checkpoints, et cetera. Now, because we started a little bit late, could I go to the next slide? I was going to tell you a little bit about neurobiology, because I don't know if you realize that yeast is a great place to study neurobiology. What does a cell do when it gets a double-strand break? So it's the neurobiology of the cell. It has to know what to do. What does it want to know? It wants to know, where did I get this break? I know that this is a complicated slide. I'm going to show it on the left side. Double-strand breaks get processed to the single-stranded DNA, and that's what elicits the checkpoint response, which stalls everything so that the cells can repair their DNA damage. Now, the interesting thing is, if, for example, a cell has an uncapped telomere, that can give rise to something that looks like a double-strand break. It has to be processed, and it elicits a checkpoint response. Next slide. Or maybe a replication fork collapses like this, and then it gets processed. It looks like a double-strand break. Again, a check. So the cell has to basically figure out where are these breaks coming from. The next part of the slide shows that it can happen from cross-linking in DNA, and the next one. Okay. So what we decided to do is we said, let's look for all the genes in yeast that make Rad52 foci show up. So we knock out a gene and we say, how did you affect Rad52 focus formation? And so that's the experiment that I'm outlining here. Now the trouble is, when you take your Rad52 plasmid and you introduce it into a diploid cell where the diploid has the gene disruption, you brought in the wild type copy of the gene so you can't measure anything. So what we did is we set up 16 separate strains, each one of which has a destabilizable centromere. We took advantage of a Carey Bloom observation that transcription through the centromere destabilizes it. So now, next slide. This blue chromosome can be destabilized, so it goes away. So now we can ask, what's the effect of this deletion on the appearance of Rad52 foci, et cetera? And so, in fact, if you go to the next slide, the cell doesn't like being 2n minus 1 for most of its chromosomes. And indeed, what it does is it endoduplicates and becomes perfectly normal. All right, so I'm going to skip all my slides now, because I know I have to be done. So go to the next slides really fast. This is what David found. David Alvaro is the student who did this. So then we wanted to look at double-strand breaks in diploids and ask really what's going on. So we took that chromosome that we can break, we made it red, and we made its homologue yellow. Next slide. And next slide, we'll skip that one. And we asked, where are they? And John Sadat told us that they're going to be in separate places, and sure enough, they are. But they move around very little. Next slide. So now what we did is we said, what happens if we make a single double-strand break in this chromosome? Next slide. And what we find is all of a sudden, the recombination center is there, but it hasn't found its homologue, or we find all 3 spots in the same place. In other words, recombination has started to take place. Next slide. And we can basically watch the chromosomes stay apart. They come together, they repair, and then they go apart again. Okay? And the Rad52 focus is there, the recombination machinery is there, and then it goes away when everything comes apart. So this is basically looking at recombination in living cells. The next slide. So what happens to the DNA? Next slide. And I forget this. Next slide, next slide, next slide, next slide, next slide, next slide. Okay. So what happens is after you make 20 double-strand breaks in a nucleus, those 2 chromosomes, which are exploring a very small amount of the nucleus, now start to explore a lot so that they can get together and repair. Okay, and that's what we found. So the next slide just shows that sometimes this, if you slow down that repair and you look after a double-strand break, nothing happens, but a little bit later, things happen. Next slide. And what happens is recombination takes place in wild type at this time, but in that delayed guy, pairing shows up later. And if you're interested in details about this, Fraulein has a poster to take a look at this meeting. Next slide, and next slide, and next slide. And how all of this works is explained by Michael Smith in his poster. Please go ahead and look at that. And the next slide just gives some conclusions about what I talked about. One is that studying yeast genetics is fun and everlasting, right? I've been doing it for over 46 years, excuse me. And labs haven't changed that much in the last 45 plus years. Well, maybe the people who work in them have, but yeast is a great system for neurobiology. Tell that to your students, the ones that all want to go off and do that. And there's still so many things to learn about recombination and every other cellular process that's very well represented at this meeting. The next slide just gives some acknowledgments. Again, some of this work I didn't talk about. Next slide. And again, there's Michael and Fraulein. And Bob Reed took advantage of these Sen strains and made some very, very nice strains. And he and Eric are going to tell you about them in Bob's talk and in Eric's poster. And I'll stop there.